The vitreous body is the clear gel that fills the space between the lens and the retina of the eyeball of humans and other vertebrates. It is often referred to as the vitreous humor or simply the vitreous structure. The vitreous humor is a transparent, colorless, gelatinous mass that fills the space in the eye between the lens and the retina. It is surrounded by a layer of collagen called vitreous membrane separating it from the rest of the eye. It makes up 4/5 of the volume of the eyeball. The vitreous humor is fluid-like near the center, and gel-like near the edges. The vitreous humor is in contact with the retina. It does not adhere to the retina, except at the optic nerve disc and the ora serrata, where the retina ends anteriorly, at the weaker band, the dorsal side of the lens. It is not connected at the macula, the area of the retina which provides finer detail and central vision. Aquaporin 4 in Muller cell in rats transports water to the vitreous body. Anatomical features. The vitreous has many anatomical landmarks, including the hyaloid membrane, Berger's space, space of Ergolet, Weger's ligament, Cloquet's canal, and the space of Marchionni. Surface features. Patella fossa, shallow saucer-like concavity anteriorly, in which the lens rests, separated by Berger's space. Ligamentum hyaloideo-capillari, Weger's ligament circular thickening of vitreous 8 to 9 mm in diameter, delineates the patella fossa. Anterior hyaloid, vitreous surface anterior to ora serrata. Continuous with and invests in the zonular fibers, and extends forward between the ciliary processes. Vitreous base, denser cortical area of vitreous. Firmly attached to the posterior 2 mm of the pars plana, and the anterior 2 to 4 mm of retina. Posterior hyaloid surface, closely applied to retinal internal limiting membrane. Firm attachment sites, along blood vessels and at sites of retinal degeneration. Space of Martagiani, a funnel-shaped space overlying the optic disc with condensed edge. Cloquet's canal, a 1-2 mm wide canal within the vitreous, from the space of Martagiani to the space of Berger, along an S-shaped course mainly below the horizontal. Internal structures of the vitreous. The vitreous body at birth is homogeneous with a finely striated pattern. With early aging the vitreous develops narrow transvitreal channels. The cortex is denser than the center with development. From adolescence, vitreous tracks form from anterior to posterior. These vitreous tracks are fine sheet-like condensations of vitreous. Named tracks. Retrolental tract, extends posteriorly from the hyaloideo-capsular ligament into central vitreous. Coronary tract, external to the retrolental tract, and excluding posteriorly from a circular zone overlying the posterior 1/3 of the ciliary processes. Median tract, extends back from a circular zone external to the coronary tract, at the anterior margin of the vitreous base. Preretinal tract, extends back from the ora serrata and vitreous base. Biochemical properties. Its composition is similar to that of the cornea, but the vitreous contains very few cells. It is composed mostly of phagocytes, which remove unwanted cellular debris in the visual field, and hyalocytes, which turn over the hyaluronan. The vitreous humor contains no blood vessels, and 98–99% of its volume is water, as opposed to only 75% in the cornea. In addition to water, the vitreous consists of salts, sugars, vitrosin, a type of collagen, a network of collagen type 2 fibrils with glycosaminoglycan, hyaluronan, opticin, and a wide array of proteins. Despite having little solid matter, the fluid is substantial enough to fill the eye and give it its spherical shape. The lens, on the other hand, is tightly packed with cells. The vitreous humor has a viscosity 2 to 4 times that of water, giving it a gelatinous consistency. It has a refractive index of 1.336. Development. The vitreous fluid is not present at birth, but found after age 4 to 5, and increases in size thereafter. Produced by cells in the non-pigmented portion of the ciliary body, the vitreous humor is derived from embryonic mesenchyme cells, which degenerate after birth. The nature and composition of the vitreous humor changes over the course of life. In adolescence, the vitreous cortex becomes more dense and vitreous tracks develop, and in adulthood, the tracks become better defined and sinuous. Central vitreous liquefies, fibrillar degeneration occurs, and the tracks break up, sine reses. Coarse strands develop with aging. The gel volume decreases with age, and the liquid volume increases. Cortex may disappear at sites, leading to liquid vitreous extruding into the potential space between vitreous cortex and retina, vitreous detachment. Clinical significance. Injury. Unlike the fluid in the frontal parts of the eye, aqueous humor, which is continuously replenished, the gel in the vitreous chamber is stagnant. Therefore, if blood, cells or other byproducts of inflammation get into the vitreous, they will remain there unless removed surgically. These are known as floaters. If the vitreous pulls away from the retina, it is known as a vitreous detachment. As the human body ages, the vitreous often liquefies and may collapse. This is more likely to occur, and occurs much earlier, in eyes that are nearsighted, myopia. It can also occur after injuries to the eye or inflammation in the eye, uveitis. The collagen fibers of the vitreous are held apart by electrical charges. With aging, these charges tend to reduce, and the fibers may clump together. Similarly, the gel may liquefy, a condition known as syneresis, allowing cells and other organic clusters to float freely within the vitreous humor. These allow floaters which are perceived in the visual field as spots or fibrous strands. Floaters are generally harmless, but the sudden onset of recurring floaters may signify a posterior vitreous detachment or other diseases of the eye. Posterior vitreous detachment. Once liquid vitreous enters the subhyaloid space between vitreous cortex and retina, it strips the vitreous cortex off the retina with each eye movement. Postmortem and forensic. After death, the vitreous resists putrefaction longer than other body fluids. The vitreous potassium concentration rises so predictably within the hours, days, and weeks after death, that vitreous potassium levels are frequently used to estimate the time since death, post-mortem interval, of a corpse. The metabolic exchange and equilibration between systemic circulation and vitreous humor is so slow that vitreous humor is sometimes the fluid of choice for post-mortem analysis of glucose levels or substances which would be more rapidly diffused, degraded, excreted, or metabolized from the general circulation.
Please subscribe and thanks for watching.